Good afternoon to everybody. Thanks to the organizers. I'm really happy to be here to share with you, on behalf of Professor Aiuti, our experience on gene therapy for ADA-SCID and its potential for the ADA2. So primary immune deficiencies have been the paradigmatic model of the feasibility and success of gene therapy. And the mainstay of the treatment is the infusion of an autologous CD34 positive cells enriched fraction that in the case of ADA-SCID is transduced with retroviral vector carrying the ADA gene. So this is a general scheme of the treatment. So as you can see, the patient is going to undergo on day -4 from gene therapy to a bone marrow harvest from which we are going to select CD34 positive cells and to send them to a GMP facility. So here, these cells are going to be engineered to be reinfused in the patient on day 0. In the meantime, for ADA-SCID patients, they are receiving a low-dose conditioning regimen with busulfan, 4 milligram per kilo into 2 consecutive days in order to make space in the bone marrow to favor the engraftment of engineered cells. And in ADA-SCID patients, we are going to stop PEG-ADA around 3 weeks before gene therapy in order to favor the selective advantage of transducer lymphocytes. And the advantages of gene therapy versus a standard allogeneic bone marrow transplantation are the lack of the immune conflict between the donor and the host without rejection and GVHD, the potential reduced toxicity versus standard bone marrow transplantation for the low-dose chemotherapy, with lower morbidity and mortality, and the possibility to exploit the natural selective advantage of engineered immune cells in primary immune deficiency in particular. And just to quickly remind you, ADA-SCID is an ultra-rare monogenic disease in which immune deficiency, infections, and autoimmunity are characteristic. And moreover, it is a metabolic disease in which the accumulation of toxic metabolites is giving rise to organ alterations in the brain, in the skeleton, in the liver. So transplantation from HLA identical sibling donor is still the treatment of choice in these patients. It's associated to an overall survival above 80% at 5 years, but unfortunately it's available just for a minority of the patients. On the other hand, MAD transplantation is associated to 67% overall survival. But this survival is decreasing, is dropping to 43% and 29% in haploidentical and mismatched unrelated donor transplantation, respectively. Another option of care is the enzyme replacement therapy with PEG-ADA that is associated to a restore of immune function in the short term, but to a decline of the immunological parameters in the medium and long term. Moreover, the B-cell compartment is not fully restored, as only the 50% of the patients are going to be weaned from immunoglobulin supplementation. And for all these reasons, ADA-SCID was considered as a right disease for which a gene therapy approach could be tried, and in 2000 in TIGET, a gene therapy protocol started with a pilot study and then a pivotal study, and then it was followed by treatment of several patients on on compassionate use program, and on 2010 the drug company GSK licensed this product in order to make it available for the majority of ADA-SCID patients missing an HLA identical sibling donor. And on May 2016, we gained the European approval of this medicinal product named Streamvelis, that is actually the first ex vivo gene therapy never approved in the world. And the preliminary results of the first 10 treated patients were published by Professor Aiuti in 2009, showing an increase of T, B, and NK lymphocytes over time, and an improvement of the immune functions. And an update about this data has been recently published. We treated 18 patients since 2000. The median age at treatment was 1.7 years, ranging between 5 months and 6.1 years. 4 patients had undergone a previous unsuccessful haploidentical transplantation, and PEG-ADA was used in 15 patients before gene therapy. So the median gene therapy dose received by the patient was 9.2 million CD34 positive cells per kilo. The majority of them received intravenous busulfan, and we tested the overall survival in this patient that resulted to be 100% in a median follow-up of roughly 7 years, ranging between 2.3 to 13.4 years for the first treated patient. And moreover, we tested the intervention-free survival that was defined as survival without transplantation nor PEG-ADA administration for more than 3 months. That resulted to be 82%. So interestingly, both these parameters are higher, exceed the 67% overall survival reported for MAD transplantation. And actually, 15 out of 18 patients remain off PEG-ADA, and among the 3 patients that required PEG-ADA readministration, 2 received bone marrow transplantation from an HLA identical sibling donor that was unavailable at the time of gene therapy. And the data show the persistence of engineered cells both in peripheral blood and bone marrow over time. Interestingly, the transduced CD3 lymphocytes are accounting to 70% at 1 year after gene therapy, confirming the selective advantage of this transduced population. There is presence of transduced B cells, while the CD15 marked cells are less, because there is no selective advantage for this population. So they are considered as a surrogate marker of the engineered CD3/4 positive cells in the bone marrow. There is an increase of ADA activity that results in good detoxification. And regarding the immune reconstitution, we observed an increase and stabilization of CD3, 4, and 8 cells in peripheral blood with improved T cell functions and decreased use of intravenous immunoglobulins. So we were able to win 12 out of 18 patients from EVIG with a positive response in terms of specific antibody production after vaccination or after infection as varicella that occurred in some of them. And we test also the rate of severe infections, observing a dramatic drop in the number and the severity of infections after gene therapy, a drop that is improving every year after transplantation. And these babies are now going to school or kindergarten, living a normal life with peers. And as a result of metabolic detoxification, the height and weight of this baby increased and were sustained long-term after gene therapy. So in summary, retroviral gene therapy for ADA SCID results in clinical benefit. We observed an overall survival of 100% in 18 patients with an intervention-free survival of 82%. We observed long-term engraftment of gene-corrected cells, immune reconstitution, and reduction in severe infection rate, a stable metabolic detoxification, and sustained growth and improved quality of life. Most severe adverse events were not related to the drug, but mainly to busulfan conditioning regimen, and importantly, we did not observe any leukemic transformation. And this is our experience, but other ADA SCID patients treated in other clinical trials with a retroviral strategy did not present safety issues of insertional mutagenesis. Actually, worldwide, more than 40 patients have been treated. However, for the safety issues occurred for other PID as WOS, CGD, and SCIDX1 with the use of retroviral vector. The search for safer vectorial strategy was mandatory, and this is the pipeline of the actual trials running at TIGET with the lentiviral vector. We started in 2010 2 trials for Wiskott-Aldrich syndrome and metachromatic leukodystrophy. In 2015, we started with beta thalassemia, and by the middle of 2017, we will start with Hurler disease. All of them have been in-licensed by GSK. So based on this background, there is a rationale for to think to gene therapy for the ADA2, because there is an unmet medical need, because there is the need for a deeper knowledge of the effect of every mutation on the clinical phenotype, because there is the need to look for definitive treatment from conservative ones, because bone marrow transplantation has been performed with encouraging results in some patients, because there is an experience and an effective result from retroviral and lentiviral gene therapy for ADA SCID, because there is a sufficient experience from lentiviral gene therapy attempted for other diseases, with the possibility to target the gene expression to specific tissues and to gain the overexpression of selected enzymes. So the project objectives would be to develop either constitutive and myeloid-specific lentiviral vectors encoding for the lacking gene, for expression in the hematopoietic stem cells or restricted to the myeloid cell lineage. We want to evaluate the efficacy of the gene transfer into hematopoietic stem cells and differentiation into monocyte and macrophages, the ability to restore ADA2 expression in human macrophages, and later we want to test these vectors in the mouse model, and we want to choose the right vector to develop toxicity, safety, and biodistribution studies in order to go to clinical translation. So there are already potential hurdles that we can recognize, as a murine counterpart of CECR1 is lacking at the moment, and there is no animal model available. So these studies will be conducted mainly in human cell lines with mutations induced by CRISPR-Cas9 system, and we know already that myeloid lineage cells, such as the monocyte-derived macrophages, are refractory to lentiviral transduction, so they will require special protocols. So we hope to give you some update about the future preclinical studies in the next years. So in the end, I want to thank all the TIGET staff, the clinical and lab staff, and our external collaborators for the ADA SCID experience. Thank you.